Okay, I'm still getting the little spinny thing. I'm not sure if it's recording or not, but
it's recording.
Okay. Well, it's still kind of spinning around and around on where I am. There. Okay. So let's go ahead and start actually looking at eukaryote diversity. So we'll go ahead and switch over to presentation. And let's go ahead and start looking at different groups of protists. And we'll eventually get to the root of the animal kingdom itself. Many of the groups we're going to talk about are not actually that closely related to animals. We know that now. There was a tendency to put them all in the kingdom Animalia, but we now know there's enormous diversity in what we used to lump together as Protista. But they're kind of still in here because we don't have any other real place to put these in the curriculum. And we'll start by going back to Carl Linnaeus, a Swedish botanist, of course. I assume you've heard of him. Remember one lecture, I said he was a Swedish botanist, and I don't know what came over me, but I picked up a couple of plant sprigs on the table and started going, "Ying more the beard nick nerd, bark, bark, bark." So here we have the classification scheme where all the living things are in the two kingdoms, the Animalia and the Plantae. Okay, sorry about that. Sorry. Come on, professional dignity, Wagner. Right. And he'd originally classified all living things into the kingdom Animalia and the kingdom Vegetabilia, which we now call kingdom Plantae. Kind of trivia, but he actually tried to extend his classification scheme to a third kingdom, the kingdom Mineralia, for rocks and minerals. He abandoned that part. He never really could get it to work right. But he put everything into two kingdoms in the final version of his classification scheme. If you moved and ate, you're an Animalia. If you don't move and you make your own food, you're in kingdom Plantae, which seems simple enough. But as light microscopes got better, it became increasingly obvious that some organisms didn't fit into either group very well. This guy, Christian Gottfried Ehrenberg, was one of the first to demonstrate that single-celled critters, these protozoans as they were called, were actually complete and integrated organisms in their own right. That they had structures that were at least comparable in some sense to organs, the things we'd now call organelles, and that they excreted and they ate and they had metabolism of some sort, that they were, they were organisms in every sense of the word, that they were completely able to sustain independent life. And some of these little organisms look animal-like and some look plant-like. Over on the left, you've got an organism called Euglena. Now, Euglena can actually swim, and it's actually capable of feeding. It's got a structure called a gullet that it can take food particles into and digest them. And you can't see it really well in this picture, but it's even got a, quote, eye. It's not really an eye, but it's a light-sensitive spot at one end. But it's still photosynthetic, so it used to get treated as a plant. A non-photosynthetic organism at the right called Astasia looks an awful lot like Euglena, but it's not photosynthetic, and it eats things. So it was treated as an animal, and yet it turns out that there's no real difference between the two, that Astasia is what you get if you take a bunch of Euglena and you grow them in darkness, What eventually happens is that the chloroplasts break down and disappear, and you end up with a colorless organism that is forced to rely on feeding. So has to eat things because it now can't photosynthesize. Other than that, they're the same thing. You could turn euglenas into astasias if you just grew them without light. So do we have one species or two? And do we have a plant or an animal? And the answer is that a lot of life goes beyond that plant-animal dichotomy, that it's not really correct to put euglena into either. A common thing to do has been to kind of put all single-celled eukaryotes into this group called the Protista. And I still use protist a lot with this meaning of a single-celled eukaryote. But if you're going to do things by common ancestry, it doesn't work. Over there on the left, you've got a less than 10 micron single-celled critter called Ochromonus. It's kind of a round cell. It's got a pair of flagella, and it's got a chloroplast with, in addition to green chlorophyll, it's got some pigments that make it look brown. On the right, you've got a 30-foot seaweed called bull kelp, genus Phaeocystus. There's lots of this off the coast of Central California, for example, and they don't look anything alike. Ochromonus is microscopic, and Phaeocystus is a 30-foot seaweed. But it turns out that in details of what the cells look like, in details of biochemistry and details of things like what pigments they use to photosynthesize, they are remarkably similar to each other. Huh, that's odd. So Ernst Haeckel, the guy I mentioned earlier that did those tree diagrams, put everything that was neither clearly a plant or clearly an animal into a kingdom called the Protista. His original kingdom Protista included the bacteria, the archaea, and the fungi as well. Now, something I won't test you on, but that you might encounter somewhere, an otherwise nearly forgotten guy by the name of Hogg had put the same organisms into the kingdom Protoctista. And I mention this because Lynn Margulis always argued that the correct name was not Protista, it was Protoctista. And so she wrote and edited whole books about the Protoctists, and all of her papers referred to Protoctists instead of protists. The thing is, this has never really caught on. If you read Margulis's own writings, of which there's a lot, or the writings of the people that were influenced by her, you will encounter the word protoctist and protoctista. Outside of that, this has never really caught on, and just about everybody says protista. So I'm just saying that in case you encounter protoctista down the road, it's the same as protista. Okay, so Haeckel creates Protista for everything that's neither a fungus or a plant. A guy named Chatton then takes out the prokaryotes and divides life into prokaryota versus eukaryota. Finally, in 1969, a guy named Whitaker takes the fungi out, and that creates a scheme that was taught a lot, at least when I was in grad school, that you may have been exposed to, called the Five Kingdoms. You have the prokaryotes, the protista, fungi, animals, and plants. I actually teach a modified version of this in my 1400 class, the Exploring Concepts in Biology class for non-majors that I teach, that I'll be teaching this second summer, by the way. But the problem with it is the protista turns out being defined by what it isn't and what it doesn't have. Protista ends up including all eukaryotes that don't have extensive cellular differentiation and don't have embryonic development. You're defined based on a negative rather than on a positive. Had a friend of mine in high school who specialized in coming up with the most pointless jokes you could imagine. And one of his masterpieces was, what does a bald eagle have in common with a brick wall? And the idea is that you rack your brains and you go, okay, I give up. What does a bald eagle have in common with a brick wall? And the answer is, neither one can drive a stick shift. I didn't say it was funny. The humor comes in the fact that you're expecting humor and you end up getting hit with that. But the idea is that classifying all of these eukaryotes in one kingdom on the basis of what they don't have makes as much sense as grouping together a bald eagle and a brick wall based on what they don't have, the ability to drive a stick shift. And the protista, as it's been defined, includes organisms that are very closely related to true animals and true plants and true fungi, plus a lot of groups that aren't anywhere close to these and don't resemble them at all and don't even resemble each other very closely. And as we have protista defined, it's paraphyletic, meaning that the last common ancestor of everything that's put in the protista was also an ancestor of animals, plants, and fungi. You probably cannot read all of that detail, and you don't really have to. This is a cladogram of eukaryotes based on a massive data set that was assembled 10 years ago. And it may seem like a mess, but it may give you some idea of just how much protist diversity there is. And I've suddenly realized that you may not be able to see animations when I'm projecting like this. But the animalia are pretty much the top half of that purple box called Opisthoconta down there at the bottom. The true plants are about 3/4 of the way up. There's a little grouping, I don't know if you can even read it, but it includes ginkgo and Arabidopsis. Those are true plants. The fungi are down at the very bottom, about the bottom third of the purple box. And everything else belongs to many branches, and many of them are not especially close to either one of those three groups. Chat me this. I don't know if you can see the animations and the colored boxes that I put in. And if you can't, then I won't include things like that in future iterations of these slides. But let's just suffice it to say that animals, fungi, and plants don't take up all that much space in this diagram that you see. Most of what you're looking at is various branches of eukaryotes that don't belong in any of them. So protista is all of that, and you can see why that's a paraphyletic group. The last common ancestor of every one of those branches was also ancestral to animals, plants, and fungi. And they're also very subject to convergent evolution. When I was in high school, we learned that the protista contain four phyla, and one of them contained all of the amoebas, and it was the phylum Sarcodina. And it turns out that protists, like on the left, you have this thing called Nuclearia, this little shapeless amoeba that looks a little like a fried egg. In the middle, you've got Acanthamoeba, which can actually make people sick. You do not want to get this thing in your eye or even in your brain, although fortunately that's rare. And over on the right is yet another amoeba species called Niglaria, which is another one that you really do not want to get in your brain. We'll talk about those later. And those three different amoebas turn out to belong to very widely separated branches on the tree of life. They're not each other's closest relatives at all, nowhere close. The latest scheme puts eukaryotes in 5 different supergroups. We're not going to cover all of these in detail. This doesn't include a few obscure taxa that we don't yet know where to put them. This is still subject to revision and modification, but it seems to work fairly well. That you have a eukaryote supergroup called the Amoebozoa, and a second one called SAR, which stands for Stramenopiles, alveolates, and Rhizarians, and a third group called the excavates. The fourth group, the Opisthoconta, includes all of the animals and all of the fungi and protists that are more closely related to them than anybody else. And then the fifth group, we're not going to talk about much. The Archaeplastida includes plants, green algae, red algae, and a couple of others. And if you want to know about Archaeplastida, we've got a perfectly serviceable botanist on the faculty, and you can go take Origin of Land Plants with Dr. Noyes and learn all you care to about them. In many cases, we can find similarities in structure that link these together. But we also define these groups by similarities in DNA sequences. And I'll walk you through all of these in turn, and I'll probably also try to type up a handout to help make all of this a little more clear. And again, we're not necessarily going to cover every one of these in detail. The Archaeplastida in particular mostly fall outside the scope of this course. So we'll start with the Amoebozoa. As I mentioned just now, amoeba shape and amoeba locomotion has evolved several times, so not all amoebas go in the Amoebozoa. Cells in the Amoebozoa don't have a fixed shape, although they may have a tendency towards taking a particular shape and size. And they move by amoeboid locomotion, duh. And in amoeboid locomotion, the cell extends one or more little extensions of the cell body called pseudopods, Greek for false feet. And an amoeba can make its cytoplasm flow into one of the pseudopods. So the amoeba will first extend a pseudopod, and then more and more of the amoeba will flow into the pseudopod until the whole amoeba is where the pseudopod is. And we repeat this process. I'll talk in a little bit as to how that works on a cellular level. It's pretty much the same way that, for example, your own white blood cells move through tissues. And by the way, one very good reason for studying invertebrate zoology is you can find connections with all other branches of biology, including health and medicine, all over the place. And one of the things that we learn from looking at invertebrates is we can find all kinds of model systems for understanding processes that have very wide application. So, for example, you can learn some things about how your white cells move through the body by studying amoeba. Another cool example is the basics of action potentials, the way that neurons send messages in this wave of membrane depolarization. Was actually worked out not in human neurons, it was worked out in giant squid. And I probably don't have to tell you how much of genetics and heredity has been worked out in fruit flies, and so on. There's lots of examples, and you'll see lots of them in this course, and there's tons more, where studying some invertebrate that you might not think is of any relevance at all can shed light on, very central biological questions that affect, our knowledge of ourselves, medicine, veterinary medicine, and things like that. So if this seems like a deadly dull march through the phyla, I'll try to show you how you can make some connections to other things. Even if you don't want to be an invertebrate zoologist, I'm assuming that at least some of you are interested in medicine or maybe dentistry or veterinary medicine, and I promise you'll find stuff that makes connections with those fields. Right, anyway. So amoebas move by amoeboid locomotion. Some members of this group are naked. They're just bare cells. But others of them have shells, and we call these testate amoebas. Testa happens to be Latin for a pot. And the shells of these testate amoebae, the shells are actually called tests. So if I put these on the exam, you're going to have tests on the tests. Right. And in some cases, the tests are made of organic matter. They may be made of silica, which is produced inside the cell, occasionally calcium carbonate, or others will pick up little mineral grains and glue them together to form a test. And we call that agglutination, when the cell glues little tiny sand grains together to make a shell for itself. So there you saw this guy in the last slide, or the last presentation. This is Amoeba proteus, your classic large amoeba. The nucleus is kind of in the center. The amoeba's moving from left to right. And down below, you can see there's a food item. I think that's a single-celled green alga. And the amoeba's reaching around to pick it up with a couple of pseudopodia, these extensions of the cell that cytoplasm can flow into. And that process of engulfing food items by surrounding them with pseudopodia is called phagocytosis. And again, it's the same way that your white blood cells destroy invading bacteria. I haven't finished it yet, but I'm actually working on a lab that would actually look at this and would make the connection a little bit more clear between your immune system and the behavior of these amoebas. So you can see several pseudopods. At the left, in this species, there's this kind of lumpy tail end. It's called a uroid. And the cell does have polarity in that it's got a front end and a back end. Even though it's otherwise completely shapeless, there's still a definable tail. They move. I'm going to have to find a video that shows this because this is a lot easier to show in living taxa than it is to explain just by bare slides. And yeah, if I had the brains the gods gave cheese, I would have set one of these videos up in advance, but I'll post one to the course playlist that I'm going to create on YouTube. What makes amoebas move is called sol gel transformation. The inner cytoplasm is very liquid, and that's called a sol. You know, think of, solution and solute and solvent and all that. Sol means the liquid cytoplasm. The outer cytoplasm in an amoeba is actually very firm, and that is because molecules of actin, a protein, it's also found in your muscle, for example. Actin molecules in the sol are not stuck together. The stuff can flow, but in the outer cytoplasm, in a state called gel, the actin molecules assemble themselves into filaments that form a rigid structure to it. So on the outside, you have this gel where the actin molecules lock together and they make everything firm. So you have firm, jello-like cytoplasm on the outside and very runny cytoplasm on the inside. The endoplasm flows forward. When it reaches the tip of a pseudopod, it kind of fountains around and, it gets squeezed through the pseudopod and then splits in either direction. And right there, it turns into gel, where those actin subunits lock together and form something solid. That process reverses at the rear, where actin joins with another protein called myosin, which is also one of the proteins found in your muscle. And the actin and myosin together contract, and that creates force that squeezes the liquid cytoplasm, the sol, forward. So the amoeba kind of acts like a giant medicine dropper, in that contractile forces at the back of it squeeze the liquid parts forward. And then at the back, the actin molecules disassemble and the cytoplasm turns back into the sol state. So you have gel on the outside. At the back, it's constantly turning into sol and getting squeezed forward. At the front, it fountains around and then turns into gel on the outside. I'm not having an easy time describing this without some video, but I promise I will put one up. Since I didn't remember to make it a part of this presentation, I will put one up for you to see how this works. A little bit of amoeba diversity. These happen to be 4 that are pretty common in fresh waters. These are all the kind of things you could conceivably find in pond water. You see they have different shapes. They've got pseudopods of different sizes. They're all about the same size in these pictures. And so, for example, acanthamoeba over there at the top left has pseudopods that are shaped like cones. And Flabellula at the top right has pseudopods that are shaped like big fans, and so on. So you can tell amoeba species apart to an extent because they tend to take very typical forms. They don't all look alike. There are amoebas that cause human disease. This one is Entamoeba histolytica. Those of you that I dragged through parasitology 50 million years ago last semester might remember these guys because they're the causative agent of amoebic dysentery. That's an infection that causes severe diarrhea. It can cause ulcers in your intestine. There are cases where this thing will eat clean through your intestine and end up causing abscesses in your liver. Parasitology people might know this, but there was a great parasitologist named Felix Schaudin. He was German, and he hypothesized that Entamoeba histolytica caused dysentery, and he proved his hypothesis by drinking a bunch of Entamoeba histolytica and coming down with dysentery. And in fact, he died from it. That's dedication right there. That is devotion to your craft. You know, confirming your hypothesis by killing yourself. Like, that's hardcore. Anyway, I digress. Entamoeba histolytica alternates between an active feeding stage called a trophozoite. Trophane is Greek for to feed. You might know it from autotroph and heterotroph, right? Self-feeder versus feeds on different things, autotroph and heterotroph. What shows up here as well, trophozoite means feeding living thing. And it alternates between trophozoites and inactive cysts. And you see one of them on the right there. And these cysts pass out of the body in feces, and those are potentially infective. If you take cysts into your body by the unfortunately named fecal oral route, which just refers to accidentally eating or drinking something that's been contaminated with feces, food prepared by somebody that didn't wash their hands, that sort of thing. The trophozoites colonize the intestinal lining. Some of those trophozoites form cysts, and the cysts reach new hosts by, again, the fecal oral route There's some evidence that in some circumstances it can be sexually transmitted as well. But yeah, good argument for washing your hands after using the bathroom. This is just another one that can cause human disease. This is a free-living aquatic amoeba that's very abundant in soil and fresh water. I've seen it in pond water samples from around here. It's called acanthamoeba. Acanthos is Greek for a spine or a thorn. So this is a thorny amoeba. And it's very hardy because it can form resistant cysts. That's one on the right. And it can go into the state of suspended animation, build a wall around itself, and then just sit there for at least months, possibly years, waiting for conditions to improve. And these cysts can blow around in the dust. They can dry out, and they can just wait for better conditions to return, and then out pops the acanthamoeba. Oh, sorry about that. I should have warned you. I forgot. I will normally try to warn you if I've got a slide that might be kind of gross. Acanthamoeba is normally harmless, but it's capable of causing infections. And one way it can happen is if you get them contaminating things like contact lens solution. If they blow into your eye, normally your tears wash them out. But if you get them in your eye and they don't get washed out, like, for example, if you keep your contact lenses in too long and you don't clean them, or if you clean them with a contaminated solution, you can get acanthamoeba causing infections of the cornea of your eye, which can cause blindness if you don't get it treated. So that is a rather nasty case of acanthamoebic keratitis. So yeah, if you wear contacts, keep those things clean, or you'll end up with that. Acanthamoeba is also of interest because it's got bacterial symbionts inside. Remember I said that endosymbiosis was all over the place in the living world? Here we go. There's a slice through an acanthamoeba, and all of those dark-colored, kind of round to polygonal things inside are a bacterium called parachlamydia, which has the potential for causing respiratory infections. There's another bacterium called Legionella that is famous for causing a mass outbreak of respiratory illness at a hotel in Philadelphia that was hosting a convention of members of the American Legion. That's a veterans group. And I can't remember how many people died, but it was pretty severe. Nobody had ever seen this before. And unfortunately, the disease is called Legionnaire's disease. It's caused by a bacterium called Legionella, and Legionella can live inside acanthamoeba. And it's thought that acanthamoeba can be a sort of Trojan horse by which bacteria like these can colonize the human body. So another thing that's interesting to study. There you've got a perfectly harmless testate amoeba called Arcella. You're seeing it kind of from above. The test from the side actually looks kind of like a big floppy beret. You know, the sort of thing you'd wear if you're an intellectual in a cafe in Paris or something like that. So the test is round and kind of flattened. It's made of some kind of organic matter. I don't think it's exactly chitin, but it is kind of similar. The main body of the cell is inside that beret, and there's an opening through which pseudopodia can extend out. So this thing kind of looks like an amoeba stuffed in a beret that's sticking out pseudopods and creeping around. And I wish I could come up with a better image than that. Think of an amoeba smoking a foul cigarette and saying, if we do the philosophy, we conclude the existentialism. That's Arcella right there. There's tests of a testate amoeba called Difflugia that shows what's called agglutinated test construction. These tests are made of little sediment grains that the thing picks up, and it glues them together in this rather orderly pattern. These have very consistent sizes and shapes. We now know, by the way, that the Amoebozoa also include most of a group called slime molds. This includes acellular slime molds like this one. This is a slime mold in genus Physarum that's basically one ginormous multinucleate amoeba, one cell with many thousands of nuclei that live in moist terrestrial habitats. This is the kind of thing that covers the undersides of logs in moist forests. There's lots of these in damp mountain forest in places like the Appalachians. So you have acellular slime molds like that. And then the cellular slime molds, like Dictyostelium, their members spend most of the time as very small free-living amoebas, crawling around and eating bacteria and actually picking up and carrying bacteria inside them. If you know Dr. Hazelcorn, she specializes in these, and she's a good person to work with, by the way. And she works on the bacteria that are found inside Dictyostelium cells and what they do. And what individual Dictyostelium cells do is when they're starved, they'll come together and join together into a millimeter-sized thing called a slug that's just made of many thousands of amoebas all crawling together. And the slug will crawl, and you're seeing developmental stages of slugs. The slug will crawl, it'll heap up, and it'll form ultimately a big ball on a stalk, kind of like a living tetherball. That ball is packed full of cells that secrete walls around themselves and become spores. And when the whole thing dries up, the spores blow away on the wind, and with any luck, some of them will drift into a better environment with food, and they'll become new amoebas again. So this thing is mostly single-celled, and then they join to make a multi-celled structure, which is a really neat trick and raises some really awesome evolutionary questions. Dr. Hazelcorn's office is right next to the classroom where I'd normally teach this, and I'd tell you all to go bother her because she's a cool person and she knows a lot about these. But since she's not here, I'll have to tell you just to do it, after we come back to campus. Anyway, so there you have cellular slime molds in the Amoebozoa. And that's all I'm going to say about the amoebas. I'm going to go ahead and I do want to, we've got about 30 minutes to go, and I do want to use our time productively. Do we have any questions right now while I'm swapping? Let me see what came up here. Okay, so you could see the color boxes and you could hear the sound effects. That's good for me to know. I think with other browsers that doesn't always work, but it's good to know that it does seem to work here. So give me a second to pull up just a few more slides. And let's see, where do I want to go from here? Oh, okay, this is going to be big. All right, I'll start sharing again and present. Okay, I've said my piece about Amoebozoa. The next really big group of eukaryotes uh, doesn't really have a proper name. It's called SAR. And SAR is made up of three main subgroups: Stramenopiles, alveolates, and Rhizaria. Oofda. Stramenopiles, I'm not going to have much occasion to talk about because most Stramenopiles are traditionally considered plant-like. And again, that's a false dichotomy, but I tend to exclude the photosynthetic protists out here just because I have to include something or the class would never end. So Stramenopiles includes some unicellular photosynthetic forms. On the left, that is a diatom. Diatoms are single-celled protists in silica shells that are photosynthetic and very, 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 very common in both the oceans and in freshwater habitats. You'll probably see plenty of diatoms in the course of this class. And then over on the right, we have a multicellular photosynthetic organism. This is bull kelp, and they're both in the Stramenopiles. They've got the same types of photosynthetic pigments, some other details of how the cell is put together that unite them. They also include many non-photosynthetic protists, some of which outwardly look like fungi. This one's probably the most famous, Phytophthora infestans, noted because it caused Angorta vor. Any of you guys know what Angorta mor is? That's Irish for the Great Hunger. Anybody know what happened in Ireland beginning in 1846? There was a mass disease of the potato crop. I've got a suspicion. I just heard the let me see. I just heard the text going off. So yeah, this was the good job, Wendy. This was the potato famine. This was a mass disease beginning in 1846 of the potato crop, and Ireland was actually raising lots of food and exporting it to England. But Ireland was full of desperately poor people whose main subsistence was potatoes. Potatoes will grow in bad soil, and they're fairly nutritious, and you can live, albeit not very well, on a potato-heavy diet. But when Phytophthora infestans infected the potato crop, potatoes rotted in the ground, and something like 1 million Irish people starved, and about another million and a half emigrated, many of them to the United States, beginning in the worst year of the famine, which people still remember as Black 47, the year 1847. Some of you might have ancestors that came to this country because of the potato blight. And that was caused by a protist that convergently looks and behaves like a fungus, although it isn't one. This is it, Phytophthora infestans. There's some other protists in this group that look superficially fungus-like. A whole group called the water molds or saprolegnias belongs in here as well. Most Stramenopiles have a flagellated stage, and if it's present, there's usually 2 flagella, and one but never both has tiny branches coming off it called mastigonemes, which means that it looks a little bit like a tiny strand of tinsel. So that's what those look like. That's a shadow cast electron micrograph right there. You see the 2 flagella. One of them is smooth, and the other one has these little tinsel hairs branching off of it. these mastigonemes. So much for the S in SAR. And I won't say much more about them. Rhizaria are mostly amoeba-like protists, but they're not closely related to the Amoebozoa. This has gone kind of convergent. Many Rhizarian groups, they don't just have pseudopods, but the pseudopods contains a specialized support structure. It's basically big bundles of microtubules. And the pseudopodia are, because they've got these microtubule bundles glomming into them, the pseudopodia are a lot firmer and much more permanent and held in place by this internal support. And when we have this, we call these axopods. Two groups of Rhizaria are very important geologically because they not only have axopods, they make skeletons. And they're some of the most abundant fossil groups that we have. Both of these have a fossil record going back over 500 million years. They're the Radiolaria and the Foraminiferida. And I won't specifically, well, I'll actually show you these in lab tomorrow, but if you want to jump the gun, your box of prepared slides should have a slide with Radiolaria and a slide of Foraminifera, and you can take those out and look at them at any time you want if you feel bored. I'll show them to you on our video link tomorrow, but if you want to, you can do this now. Radiolaria, or as they're just commonly called, rads, have internal skeletons made of silica, aside from one little group that just has to be different and makes them out of strontium sulfate, of all things. And these internal skeletons form on a membrane structure called a capsule that separates the cytoplasm into an inner and outer part, endoplasm and ectoplasm. And I'll show you this in just a second. Forams, or Foraminiferida, the pseudopods form a network. They split and they join, and they create something like a living net. This is said to be reticulation. Reticulum is Latin for little net. Most forams, there are a few naked ones, but most forams form tests, there's that word again, that can be made of organic matter or mineral grains glued together, or most commonly calcium carbonate. And the test can have very complicated shapes. I'll show you these in just a bit. And then finally, the ones that don't belong in either the rads or the forams are sometimes called cercozoa. And I'll show you a few pictures of those in just a bit. In fact, I'll show you one now. There's a cercozoan. The common name for these is heliozoa, sun animal, because they look exactly like the drawings of the sun that you made when you were in first grade, right? A circle and then a bunch of lines radiating out. That's what these look like. And instead of pseudopods, they have these very slender hair-looking things sticking out. They're not flagella because they don't beat back and forth. They're extensions of the cell that are supported internally by these bundles of microtubules. And this particular species also happens to have endosymbionts, because I cannot pass up a chance to point out that endosymbiosis is all over the place. Because, you know, this is me. There's a slice through an axopod in cross-section, and each one of those little circles that makes this kind of double spiral shape is one microtubule. These can bend to some extent. Some organisms can actually flex them enough that they can kind of walk or roll on their axopods. And they're also used to trap food particles. There are some testate amoebas that are in this group, Cercozoa, and they're not that closely related to the other testate amoebas that we just saw. And many of them have tests made of silica scales that are secreted internally. And this euglypha on the bottom picture, you should be able to see that the test is made up of these very regular overlapping scales, very regular shape, kind of a neat trick for something that's maybe 50 microns long. If I can get into the greenhouse, I'll see if they're still there. There used to be the water cooler in the greenhouse had algae growing in it, and the algae was eaten by this very nice population of euglypha. So we used to have these live, easily available. If I can, I'll try to, and if it's still there, it may not be, but I'll try to bring some of these in tomorrow for us to look at because they're cool, or I think they're cool. And I'm the professor, so yeah. And then other cercozoans range from tiny flagellated protists to this really weird thing that is it's actually got the remains of green eukaryotic algae embedded in its own tissues. So it's got a symbiont with symbionts inside it, and it grows like this weird network of cells. It's called Chlorarachnion reptans, and it's really strange, and nobody knows anything about it, but it's an example of cercozoan diversity right there, and that's all I'm going to say about it. For the record, for the exams, you're not going to need to know genera. A little bit cooler, there's a living rad, or radiolarian. On the inside of the cell, you have most of the organelles. That's a dark endoplasm. And then in the outside of the cell, the ectoplasm is full of bubbles. It's almost frothy. You know, this is almost like a spherical cappuccino with the froth on the outside and the dense stuff on the inside. And the two are separated by a membrane structure called a capsule. And then surrounding the whole thing, you can see lots of little pseudopods, or lots of little axopods, these ray-like extensions of the cell. And you can also see some spines that are part of the skeleton. And the skeleton looks like this. It forms right on the capsule, aside from these little spikes that stick out. And they've got this very beautiful 3D structure to them. If they were bigger, people would harvest these for Christmas tree ornaments. Again, a very sophisticated skeleton to make for a single-celled organism. This is pretty cool stuff. Many of them are spherically symmetrical, but there's one major group within the rads where the skeleton is typically conical or sometimes said to be helmet-shaped. So there you've got a conical one on the right. The one on the left, I guess you could wear that as a helmet. It looks like the kind of thing the orcs would wear in Lord of the Rings, if you ask me. But there you have it. They're not all spherical. Some of them are cones. Rads, by the way, you're likeliest to see out in the open ocean. They float. They live out there. Only in the ocean. There aren't any freshwater radiolarians that I know of. So a lot of people have never seen them live. You kind of have to be like off on an oceanographic research vessel to get a good look at the living ones. But the skeletons can be very common in certain bodies of rock. And the same is true for the skeletons of forams. You have a test in the center. That brown thing is, you know, the shell of the foram. And then coming out of that, you have this huge network of very fine reticulopodia. And remember, reticulum means a net. You can see where these branch and they rejoin and branch again. And so you have this net. Some of these have endosymbionts and others trap smaller organisms inside and digest them inside the cell. using that reticulopod as a, well, as a trap net. And foram tests can take on a lot of diverse shapes. There's some where the test is just one chamber, but most of them have many chambers. And sometimes the chambers make a spiral shape that superficially looks like a snail, although it's not. They can take in various other shapes like that. Most species, it's calcium carbonate. There's some that make organic tests. And again, if you feel like you've got nothing better to do, you should have a prepared slide of forams and one of rads that you can look at with the microscope that you should have by now. Foraminifera means thing that carries foramina, and a foramen is a pore, a little hole. Your pre-meds, you're going to have to learn the foramina of the base of the skull that the cranial nerves pass through. You've got the foramen ovale and the foramen lacerum and the foramen this and that. I can't even remember all of them now, but it's the same word for a small hole for a nerve to pass as it is here. The foramina are these little pores that make the whole thing look like it's been shot with buckshot. And the reticulopods pass out of those pores and then join together and make that network. Lots of forams are present in sediments. They can be very common in tropical seas. There are some that float, and in this floating planktonic foram, the reticulopods act kind of like the fluff on a dandelion seed. They trap food particles, and they also keep it from sinking. They slow down the sinking rate greatly, which is a good thing because the foram can't swim. It can't, if it sinks, it can't push itself back up. This way it can just drift on currents for very long periods and stay up in the water column its entire life. And these are so abundant in rock deposits because they're constantly living, dying. They're about the size of sand grains. You can see most forams with the naked eye, and they can build up over many years in very thick layers on the ocean floor, and those layers can harden into limestone. These are some fossil forams in the genus Nummulites. They're easily big enough to see with the naked eye. They're roughly, I'd say maybe 5 to 10 millimeters across, sometimes more than that. Nummulites is Greek for little coin, if that tells you anything. They can be as big as a dime. And there's limestone in Egypt that's made up mostly of Nummulites, 40 million years old. This is what makes up the bulk of the pyramids of Egypt. The pyramids are made of forams. The pyramids, be it said, used to have polished marble on the outside, but the inside of the pyramids was mostly much cheaper limestone, and that limestone is made up mostly of Nummulites forams. There are plenty of rock deposits on Earth that are made up almost entirely of forams or of rads. One of my fellow grad students was studying the fossil rads to be found in the rock that makes up the bedrock of San Francisco. And there are sizable portions of the deep ocean floor today covered in mud made up of dead foram tests. It's called foram ooze. This is what it looks like, and it could someday harden into limestone. And then in 50 million years, somebody might make pyramids out of it because, you know, why not? In fact, forams are so awesome that the next time you're in Hong Kong, you can go visit this park that's filled with sculptures of tiny forams built by a local marine biologist who convinced the local authorities to build this. So every one of these funny looking sculptures is actually a giant model of a different foram species. So yeah, instead of going birding, you could go faramming. Okay, 12:02. I'll theoretically should keep going for another 8 minutes to make sure you're getting your money's worth, but I'll go ahead and end it there for the day. I'll turn off presenting and turn off recording.